Okay, thank you all for coming. We'll go ahead and get started. So it's my pleasure today to introduce our speaker, Dr. Jonathan Stillman. Jonathan is a professor at San Francisco State University and an adjunct professor at University of California, Berkeley. Jonathan grew up in Minnesota and he received his BS in biology from the University of Minnesota. He then earned his PhD with George Somro, starting at Oregon State University, which is where I first met Jonathan in grad school. And then Jonathan moved to Stanford University when George shifted his lab group to Hopkins Marine Station. So Jonathan's now been a professor at San Francisco State University for the past 20 years, where he's a marine environmental physiologist. And there are at least three things that have long impressed me about Jonathan's research over the years. I think first, Jonathan's research is deeply integrative, so his lab uses approaches ranging from field studies to traditional physiological measurements to transcriptomics and functional genomics, and I think this integration of different approaches is a real strength of his lab group. Second, Jonathan's research in his lab is always really grounded in a deep understanding of the environmental conditions that organisms see actually out in the real world. So ever since Jonathan was a grad student, he's been a real leader among physiologists in measuring the actual temperatures that organisms experience out in their natural habitats. And as a result, I think Jonathan's research provides unusually robust insights into how variation in habitat temperatures shapes the physiology of organisms and their responses to climate change. And then lastly, Jonathan has a deep appreciation for natural history and organismal biology, and especially for crabs. So when Jonathan was a prospective grad student and he was first reaching out to his future advisor, George Somro, Jonathan's inquiry letter expressed that he really just liked crabs and wanted to study crabs in grad school. And so Jonathan has maintained, isn't that true? That's true, right? But is that not true? I don't have a copy of the letter, but that's something that I really appreciate about Jonathan, is that throughout his career, he's really maintained this appreciation and genuine curiosity for marine creatures, especially marine invertebrates. And as someone who teaches invertebrate zoology, I appreciate that that's part of Jonathan's scientific motivation, that he hasn't lost that real curiosity for organismal biology. So it's a real pleasure to have Jonathan here at BML today, and the title of his seminar. Oh, and also, as I mentioned, after the seminar, Jonathan will be playing a Swiss Alphorn, which is here just so people believe me. So I'm sure most of you are here for Jonathan's science, but I'm sure my email about Jonathan playing the Swiss Alphorn also encouraged some people to come. So how many people are excited about the Swiss Alphorn performance after? Yeah, see? So immediately after the question and answer, we will either go out in the great hallway or just outside, wherever the acoustics are best, and Jonathan will provide a short performance of the Swiss Alphorn. But let's start with the science, and so it's my pleasure to turn it over to Jonathan, who will be speaking about, does the thermal environment induce intergenerational plasticity in the porcelain crab, Petrolistes syntypes?
Okay. Hopefully that's going to work and everyone can hear me. Put this one in my pocket. Thank you, Eric, for that lovely, lovely introduction. I can't wait till I get to introduce you someday. That's going to be so much fun. So you might, if you're a stickler for details, recognize that my title on this slide is just slightly different than the title that was advertised, in that I changed this. Is this a laser pointer also? There we go. I changed this word from transgenerational to intergenerational, and I did that for two reasons. One is that my grant office told me I couldn't use trans in my upcoming NSF proposal, in which I had written transgenerational a lot of times, so we thought intergenerational. And it turns out it's a more accurate term for what I'm going to tell you, because intergenerational means between two generations, like a parent to a child, and transgenerational means more, like a parent to a grandchild. And what I'm going to tell you about really is most accurately representing intergenerational, and it's about whether the thermal history experienced by parents influences the physiology and response to temperature in the offspring. Very Lamarckian ideas that we're going to be hearing about today about modes of adaptive evolution. And today I'm going to share data from 5 of the previous students from my lab: Alexis Hooper, Lily Pruitt, Joe Holdreith, Raphael Leon, and Max Hyper. These 4 were all master's students in my lab, and Max was a Berkeley undergraduate who worked in my lab. Many other students, including one here in the audience, have worked in my lab doing wonderful things, but I'm going to restrict the copious quantities of data I'm going to show you today to these 5 students. So I grew up in the 1970s and 1980s, and I remember summer as being a time when it was really pleasant with only a few days that were really uncomfortably hot. And from this animation, which was supposed to loop multiple times, sorry, it didn't, what we've noticed is that our distribution of summer maximum temperatures here in the Northern Hemisphere has shifted dramatically during those subsequent decades. And now we have a very high percentage of temperatures that are extremely hot compared to only a small fraction of temperatures that were extremely hot in the past. And those temperatures, of course, are influencing places across the Northern Hemisphere, but they also influence the sorts of thermal exposures that organisms experience on our coast. And here's a spot on our coast where I know a number of you routinely go, up on the Sonoma coast at Fort Ross. So this is the field site that I'm going to be talking about today. And Fort Ross is very often warm and sunny, and the organisms that live there experience warm temperatures, as I'll show you. So here we are at the field site with Lily, and under all these stones in the intertidal zone here, it's about mid-tide in this picture, are these crabs, porcelain crab, Tetralisthes cinctipes, which lives in the middle intertidal zone, and there's a congeneric species or 2 that live further down on the shore. But we're just going to talk about cinctipes today. That's my hand, same wedding band. So these are not big animals, but they're very abundant down on the shore. Is this microphone loud enough, do you think? It's going to be okay? All right. Hopefully the Zoom people can hear me. So as Eric mentioned, I do research that's grounded in understanding the natural habitat, and we wanted to understand just what the thermal environment was like under these stones. And starting when I was a graduate student, I would go out with a thermocouple thermometer and shove it under a rock as the tide receded and measure the temperatures until the tide came up. But then we decided we could do it better, and we planted dozens of I-button temperature data loggers underneath stones that were kind of higher on the shore in what we called the high intertidal zone, and lower on the shore in what we called the mid intertidal zone, but they're really all within what we would normally say is the mid zone, which is an area that is exposed to air on most low tides. And here's what we found over the course of a year, or a little bit more than a year, omitting the winter data where the rocks got rolled around. So we wanted to know what the temperatures were like underneath these rocks where the porcelain crabs lived. And to our surprise, we saw quite a lot of days where the temperatures higher on the shore exceeded 30 degrees C, and sometimes approached even 40 degrees C. But the mean temperatures, which are these thick blue and red lines in the 2 zones, were about the same. So animals living higher on the shore see much greater range of temperatures, and sometimes temperatures that reach physiologically stressful levels, whereas the mean temperatures are really the same. And so these extreme hot days The extreme hot days in the field are what we're interested in this context of climate change where we have more extreme hot weather. And this graph over here on the right, it's a little complicated. It shows the intensity of a heat wave on the y-axis and the duration of a heat wave on the x-axis in terms of days. And the size of the dots indicate the geographic area over which the heat wave occurs. And the color of the dots represent when in the 21st century these heat waves are occurring, whether it's in the middle part of the 21st century from years 2021 to 2050, or in the last 3 decades of the 21st century, and whether it's a heat wave that's likely to be very common in the 10th quartile versus very uncommon in the 90th quartile. So the dark purple ones are the ones that represent the most extreme heat waves, the ones that are likely only going to be happening a small fraction of the time. And what we see is this red dot right here. That red dot represents real data from 2003, from the massive heat wave that hit continental Europe, killed 70,000 or more people in France from excess heat. And what we can see is that whether CO2 peaks in 2040 or whether we continue in a business as usual schedule, by the end of the 21st century, we're going to have heat waves at least that big or possibly much bigger in terms of their intensity, their duration, and their geographic area. That is to say, summer is going to continue to be a hotter and hotter place in terms of daily extreme temperatures. So not only in the intertidal zone, not only will this impact the temperature, but also has the potential to impact the pH of the environment that the animals are living in. So over here we have a cartoon of temperature across time, and as the tides go in and out, we get thermal variation. And then over here, we have pH over time. And likewise, as the tides go in and out, we can get pH variation. And we think that this temperature variation will be exacerbated by warming temperatures in the future because we have warmer air temperatures, warmer water temperatures, and a greater frequency of heat waves. And we think that the pH variation can also become exacerbated into the future because we may have stronger upwelling because of stronger thermal gradients between the coast and the Central Valley. We may have acidification of the seawater because of long-term CO2 forcing, but also increased tide pool respiration because, or nearshore respiration because it's warmer and respiration rates are typically higher when the water is warmer. And those things will drive pH down. We were very interested in looking at how pH and temperature extremes influence these crabs because of their life cycle, where some of the time they would be experiencing these coastal fluctuations and other times they would be experiencing more of a constant environment. So the crabs as adults, this one is from Chile, this particular crab, in case you're wondering why it doesn't look like Petrolisthes cinctipes, is not. But the adults live in the intertidal zone, and then they go through gametogenesis, and embryos hatch out into larvae in the intertidal zone, and then they spend some time in the offshore plankton as larvae. They can spend several months as larvae, during which time they're in a constant thermal environment, and then they settle back into the intertidal zone. They settle gregariously, so they only settle where there's a conspecific adult, and they spend the rest of their life there growing up and maturing. So not only are the adults in the intertidal zone, but also the embryos that they carry are in the intertidal zone where they're developing. Today, I'm going to tell you a story about the embryos, and we're mostly going to be focusing on the embryos. I'm not going to tell you the story of the adults or the larvae or the juveniles today, but we have stories about them also for another day. And so what are the embryos doing? The embryos are extruded by the female as a fertilized egg. So the females mate, they store the sperm from the male in a little pouch inside their body, and as they oviposit the eggs, they go past that little pouch and they get some sperm, and we have syngamy, and so when those eggs are then brooded by the female, they're embryos that are in some stage of development. And in the first week of development, they're pretty undifferentiated from kind of a gross anatomical sense. And we start to see the yolk, that this red color, start to get less and less as time goes by. Around the 5th week, we see an eye spot develop. And by the time they're ready to hatch, the yolk is pretty much all gone, and then they hatch out as a larva. So during this embryonic development process, they're using this yolk. That's their source of energy. They're growing. We can see that these scale bars, at least for all the embryos, are the same size. So the embryos are getting bigger over time. And so the rate at which they use the yolk and the rate at which they grow are interesting potential phenotypes. And we can grow these embryos very easily in the lab in individual 96-well plates, and we can study individuals throughout their development and ask how they do. The first experiment that we did on the embryos was to look at interactive effects of temperature and pH. And this experiment happened actually in England at the University of Plymouth, and it was with a porcelain crab species that was in the intertidal zone, but a different one than we have on our coast here. But it led to all the research that I'm going to tell you about, so I thought I should tell you about this experiment. We took 6 crabs, 3 that we collected in May, and 3 that we collected in July. We could only really handle the embryos from 3 crabs at a time because of the infrastructure of this lab. And then we split up 192 embryos from each female. 96 of them were raised at pH 8, and 96 were raised at pH 7.6, different pHs. Those were constant pHs. And then of those 96, we took 24 of them and gave them no heat shock. 24 got an early heat shock, which is 1 hour at 31 degrees C, so immediately upon collection as the undifferentiated embryo. 24 of them got a late heat shock, which was when we saw the eye spot start to develop. And then 24 of them got heat shock both times, okay? And then we just kept them in these 96-well plates, and we looked, in this case, to see how they survived. And so here are the data for just pH 8, showing the survival probability versus time, about a month worth of time. And we see, not surprisingly, that the ones that got heat shock are surviving the least well compared to the ones that weren't heat shocked. And when we layered pH on top of this, we see that pH didn't make a very big difference. They're pretty good probably at internal pH regulation. So there was no big surprises there. But on the next slide is where the big surprise comes. I told you we collected 6 females, 3 in May and 3 in July. And here's the 3 in May, and here's the 3 in July. So in May, we saw almost no effect of any of the treatments that was consistent in terms of their sensitivity to the environment. Whereas in July, we saw heat shock have a really big impact with a big separation between the blue line and the reddish lines in each case. That means that in July, the embryos were very sensitive to the environmental stress, and in May, they weren't. So why was that? Was it because we picked up randomly in May 3 females that happened to have the right genotype to make embryos that were really robust to environmental stress, and in July, just by chance, we happened to pick up 3 females that would make embryos that were really sensitive? Could be. Was it because something about the environment that the females were doing their gametogenesis in, that is the time before they oviposited in May and the time before they oviposited in July, did that have an influence on how the females provided to the oocytes? That is, did they put different proteins in the oocytes, different RNAs in the oocytes to change the physiology of the oocytes? And that could be the case. And so this was fascinating to me, and I really wanted to try to understand it better. Unfortunately, or maybe fortunately, I couldn't spend the rest of my life in Plymouth, England. We had already been there for 3 months, and it was time to come home. And so we tried to do some of the same things at home. And we had some females that were from the field. Those are the ones with black X's, field broods. And we had some that were from the lab, the purple broods. And what you might notice, so these are the same sort of survival curves over time, but now we just have control and heat shock, not all the various different types of heat shock. It's just the early heat shock, 1 hour at a temperature that's just under the LT50 for these embryos, so lethal temperature, a little bit under that. And what we saw was quite a lot of variation in the field clot samples, here and here. Sometimes they weren't sensitive, sometimes they were sensitive. In the lab held, there was a lot more consistency. There was not really a big difference between the different females. And these are just some of the data. And so that would suggest that variation in the field, which is a more heterogeneous environment, might have led to more diversity in terms of the sensitivity of the embryos. And so we wanted to investigate that more. We also, by the way, saw the same thing with pH. So here is data for heart rate and respiration rate of embryos as a function of exposure to low pH. And this is for Syncytipes, our species here. And so if the dot is at this red line of 0, it means that pH didn't have an effect. And if it's below, it means pH slowed them down. And if it's above, it means pH sped them up. So, and the different dots are for different broods. So sometimes broods were very much slowed by low pH, and sometimes they were even accelerated by low pH. But most importantly, there was a lot of variation from brood to brood in terms of how they responded. If you ask me, how do porcelain crab embryos respond to low pH? I would say it depends. Depends on what brood they're from, right? But the question is why? And so the hypothesis is that part of the why could be because of genetic variation among parents, which is a fixed difference, but it could be because of these intergenerational effects, which are a plastic difference. It could be also transgenerational, but I'm going to use inter because it's the new era now. So just between one and the next generation. Hypothesis, pretty simple. Intergenerational plasticity exists, and that the thermal habitat during gametogenesis, the time when the ovaries are maturing all of those oocytes into mature eggs, that alters the parental contributions to the gametes, that shifts the offspring physiological phenotypes. And I'm going to show you some studies where we've investigated this in the lab to look at seasonal acclimatization, in the field to look at seasonal acclimatization, and the lab to look at acclimation to different experimental conditions. And the types of data I'm going to show you are on energetics like growth rates, yolk consumption rates, metabolic rates, hatching rates, as well as some cellular phenotypes like shifts in protein levels and mRNA levels. So hopefully you'll get that kind of interdisciplinary integrative picture that Eric was telling you you would get. Okay, so we're going to start with a lab study. So in the lab study, we went out in October 2023 during the period of the year when the crabs are not typically reproductive, at least here on the Sonoma coast. And we brought them in the lab, and we held them in the lab for quite a long time, more than a year. And they were in these cages. They're just chunks of PVC with a bunch of holes drilled and caps on each end. And each cage had a female and a male in it. And they were held under one of 4 conditions. They either never got a heat shock, or they got a heat shock once every other week, or they got a heat shock once every week, or they got a heat shock 3 times a week. And during the heat shock, my grad student Lily took these cages and brought them up to the lab and put them in a cooler that was above some water, and she heated the water to a high enough temperature so that inside the cages it was 26 degrees C for an hour. That's a temperature that's we know from previous studies is warm enough to induce a mild cellular stress response. So we know we're giving them a mild heat shock during this time. It's like a hot low tide period for these crabs, but it's not enough to kill them even if they see it 3 times a week. And then we had females, so each one of these cages has a genotype in it, right? It has a male-female pair, so their genotype is constant. And sometimes these females laid multiple clutches. So in a lot of these cages, you see blue dots and red dots. That means that there was a first clutch, the blue dot, and the second clutch, the red dot. And some females, this one and another one that's not on this chart, made a third clutch, which is pretty exciting. When we kept these females through the next season, we had some that made another clutch. Unfortunately, the data from first to second clutch is not as strong as we would like. We had a technical problem in the lab that resulted in the loss of quite a lot of data. And it had to do with a very simple thing, with students not realizing that you can't reuse the little sterile filters that go on the ends of syringes. And they thought they were being good and being conservative and reusing this because water could still go through it. And I didn't realize what they were doing, and so their water wasn't actually sterile. And they had lots of epibiotic growth and lost a lot of these clutches data. So unfortunately, we don't have as much of that as we would like. But the data I'm going to show you are hopefully strong enough to tell this story. We also had a problem. This is the other graph where you can see the third clutch of this female here. We also had a big problem with a lot of mortality, where the colored lines changed to black was where there was a lot of mortality. And a lot of it happened in this period here. And it was because we were in a shared aquarium room and someone else brought in a whole bunch of sea stars, and we had a huge nutrient spike, which resulted in a lot of mortality. So we lost a lot of our parents also from this study. The procedure generally was, as soon as we saw the embryos, we would take them off the female, and we would put them in a dish and sort of sort them out. Some of them would get a 1-hour heat shock. Some of them would stay at control conditions, and then we would raise them up through development, or we would freeze them immediately for doing the omics work. And so the omics work was done on very early stage embryos, whereas the energetics is done throughout their developmental period. Okay, so the first data are on hatching success. Here's a successful hatch, and here's a failed hatch. And there's lots of ways in which hatches fail, right? But we wanted to know just how many of them make a zoea larvae that looks reasonable. And here are the data. So here's the hatching success rate on the y-axis, and here's the parental treatment on the low axis, no heat shock to high levels of heat shock. And the colors represent the embryo treatment, whether the embryos were heat shocked or not. So if we just look at the control embryo treatment, these are the embryos from parents that had no heat shock. These are the embryos from parents that had the highest level of heat shock. We see that hatching success rates were sort of in the 50 to like 65% rate in general, with one brood that gave us 100% hatching success. Maybe there's a slight downward trend here. The interesting thing is when we look at the response to heat shock had a huge effect on the embryos from parents that were under the control treatment, but a much less effect in the embryos that were from parents that had been receiving a lot of heat stress, suggesting that the heat stress the parents received influenced the tolerance to heat stress of their offspring, which is our hypothesis, right? So these data support our hypothesis. And I love this graph. Hopefully it'll get published someday soon. I told you that we were studying the energetics. So we took lots of pictures of the embryos. So here's an embryo under our microscope. And we have an ImageJ script that works, although sometimes it takes a little bit of effort to get it to work. Has to do with how you save the files mostly. But it extracts the whole embryo as well as just the pixels containing yolk, plus this little red spot right here that it can't differentiate from yolk, but that isn't yolk. And you get the area of the embryo and the area of the yolk in terms of pixels, and you can use that to make lots and lots of graphs like these. Here, each graph represents one embryo, and so here, for example, this little embryo here got photographed 4 times during its development, and its yolk area went down, and that same embryo here grew during that period of time. And so the slopes of those lines represent the yolk consumption rate and the growth rate for these embryos. Here are the yolk consumption rates. The graph has the same structure as before. We see not a big difference in embryos from parents that were raised under control conditions. That is to say, not a big difference between embryos that were heat shocked or not. But a big difference between embryos that were heat shocked in parents that were from high thermal stress environments, where there was a lot more variation in total and a strong decline in yolk consumption rates after heat shock. Here are growth rates. And so we saw in general that when parental levels of heat stress went up, growth rates generally went down, with perhaps a strong secondary effect of heat shock to the embryo, but that wasn't the case in the medium treatment. We only saw that in the low and high treatment. So maybe a little bit less of a strong statement about the truth of that response. Okay, so overall from our lab studies, there's some strong evidence for intergenerational plasticity. Now let's go into the field. Here's our field site on a not hot day. I'm sure you've experienced the coast. Like on a day like today, it gets hot. On a day like this, it doesn't. And we wanted to know whether there was some pattern of acclimatization to embryos that were from gametes produced during the winter season, when we have the least chance of having stressful temperatures, versus the summer, when the gametes would have been made during the spring, where they have the maximal chance of extreme high temperatures. And I'm showing you quite a lot of data on this slide. Here's hatching success, here's growth rates, here's yolk consumption rates, here's respiration rates. The embryos that were not heat shocked are in blue, and the embryos that were heat shocked are in red. There was no difference in hatching success between summer and winter. Over here is summer, over here is winter. But we saw a strong decrease in growth rate. The summer acclimatized embryos grew faster in general, but with a lot of variation compared to the winter collected embryos, although no impact of heat shock to the embryos. Yolk consumption was also highly variable in the summer compared to the winter, and here we saw an increase in yolk consumption rates in winter acclimatized crabs as a result of this heat shock to the embryos. And respiration rates are where we saw the biggest effect of heat shock. The respiration rates did increase dramatically in response to heat shock, both in summer and winter. I didn't mention how we collected, excuse me, you shouldn't chew ice when you're on a microphone. Even though I lived in Europe 5 years, I still chew ice, which is a terrible American habit that no one else in the world does. How we measured respiration rates was cool. We developed a new method for this using little tiny fluorescent nanoparticles from a company called Pyrosciences. And we put them in the wells of a 96-well plate along with the embryo, and then seal them and put them in a fluorescence plate reader that has temperature control. And so it's like having 96 little respirometers. We tried doing it with 384-well plates, and it worked really well, but it's really hard to get the embryos out afterwards. And we wanted to get them out and put them back in our culture incubators, so we decided to stick with 96-well plates. Oops. That's the result right there. In terms of other sorts of traits, we didn't see a whole lot of variation. Here's the mass of the brood in the summer and the winter. Now on the x-axis are all the individual moms or mothers that we collected the embryos from. And here's the size of the mothers. So it wasn't like there was a difference in the overall fecundity or the sizes of the moms. There was some variation, but it was similar across the 2 seasons. However, we did see some sort of consistent differences. When we look at the starting percentage of yolk in the embryo and the starting size of the embryo, and this, on this graph, you can really kind of get a handle on how much data we have, because each dot represents an individual embryo that we studied. And these are all the embryos from the summer, and the blue ones are all the embryos from the winter. And now you can really see that there's a difference in these 2 populations in terms of the amount of yolk they had per body size. The ones in the winter that we studied started with more yolk for a certain body size. And you could ask, why would there be a variation in yolk? Why didn't they all have 100% yolk when you started, right? And that's because we didn't always find the embryos right after they were extruded. There was some variation. Sometimes we found embryos in week 1 and they had 100% yolk. And sometimes we found embryos a little bit later. And so they had less yolk in them. But even so, for a common starting size of the embryo, we can see that there's more yolk generally in the winter than in the summer. So they were generally larger with more yolk. We also found a seasonal difference between the mass of the brood and the carapace width of the female that had a seasonal component to it. Here the solid line is for summer and the dotted line is for winter. And the size of the dots represents the size of the starting size of the embryo, and the color represents the starting yolk percentage. And this is just to say that there were some overall differences between the morphology in the summer and winter collected embryos. We did have some evidence to suggest intergenerational plasticity here, but there's a problem, is that we collected embryos from the moms in one season, but they weren't the same moms from the ones in the next season, right? We went out and collected from all of these moms in the winter, HZW1, blah, 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 and all these moms in the summer. And so that was a problem with this study that we're trying to fix. And the second problem was that it turns out that getting those week 1 embryos was also a problem because we don't live in the field, right? We only go on certain days when it's convenient for our schedules. Maybe here at Bodega, you have better access to the field and you can go every day, but when it's 2 hours to your field site from San Francisco, you go once in a while. And I didn't point out all these data over here, but this is just the same thing I've already showed you, but now per mom instead of per season. So we started to try to put moms in cages, moms and dads together in cages just like the ones we used in the lab, and we used field ecology Z-Spar and field ecology grade zip ties to try to put these cages underneath the rocks where the crabs live. And each cage also had a little field ecology grade temperature data logger in them. And just like all field ecology grade things, they often disappeared. And so unfortunately, we don't have a ton of data, but over here are some temperature data from these cages just showing that we actually did record the temperature. And we have had some success at keeping moms and dads in the cage alive for some time, but we need to work more on this in terms of attaching them in a way that's permanent. We definitely have lost quite a lot of cages and quite a lot of expensive data loggers doing it. The next steps for our study in the lab are to use this aquarium setup that we now have. And this aquarium setup basically has 16 tanks, 8 here and 8 on the other side. And each tank has tides. So here is the high tide level, and down here at this PVC or just below it is the low tide level. And there's heat lamps, and under these heat lamps are cylinders, and the cylinders contain little crabs, and we put food in the cylinders, which is why they kind of have a greeny color right now. And to keep them alive, with different levels of thermal variation. A computer controls these heat lamps, and a computer controls the water height in these tanks. And so we can simulate whatever types of low tide heating scenarios we want with this setup. And it works okay, but it's recirculated seawater, artificial seawater, and it's no mimic for natural seawater. So if it were at Bodega, I'm sure it would work awesome. At San Francisco State, we'll see if we can get it to really work well. But I have high hopes. Here's some temperature data from those tanks, just from recently, showing what it looks like inside of a cylinder that's getting a heat spike. The blue is a heat spike every day. The orange is a heat spike just on this day. And then the red color is no heat spike. We still get a little thermal variation even when there's no heat spike, because during low tide, the room is not as cold as the water temperature is. So that's basically room temperature there. And this is just kind of a cartoon that shows the heat lamps and the cylinders and the temperatures. Okay, and so we want to do some higher resolution field sampling also. Maybe instead of going once a month, we can go every week. And we can pay attention to other things about the microhabitat, like tidal elevation. And we can also do a better job of bringing out a field portable microscope to make sure that the embryos that we're getting are early stage. And so we'll continue those same sorts of physiological assays on the broods that I've already showed you. Currently, what we're doing that I want to show you a little bit of is leverage the genomic resources that we've already developed for the porcelain crabs. We've sequenced the genome of Petrolisthes cinctipes and Petrolisthes manamaculus. And you can go dig up that genome yourself. It's published from the University of Basel, because that's where I was located when we did the genome sequencing, and a PhD student there and a postdoc there and I worked together on the assembly and writing the paper. So that name is what's on the submission, but in case you're wondering why it doesn't say San Francisco State, that's why. And here's the paper in G3 in 2023. And so Max Piper, the Berkeley undergraduate I mentioned, wanted to do an RNA-seq study. And so I said, well, that's cool. Why don't we take embryos from broods that we know from the physiological assays to be sensitive to heat stress, and embryos from broods that we know to be robust from heat stress, and do RNA-seq on them. And so Max found those samples in the freezer, and it turned out that the starting age at the time of those studies varied quite a bit, from embryos that were in what we called week 1 of development to embryos that were in week 7 of development. And so over here, you see these names, 1S, 1S, 1.5R, 1.5R. That means the week and whether they were sensitive or robust to the heat stress. And all the way to week 7. And so he submitted all those samples for sequencing, and I looked through the data for transcripts that were significantly different in their levels across all those samples. And those are the over 2,000 different genes that you see here, where each column on this heat map is a different transcript. And so it's over 2,000. There's about 400 that were either strongly upregulated in week 1 or week 1.5. Those are these 4 up here. And these, for the most part, these transcripts are not strongly expressed after that. And then there's, a couple thousand transcripts that were only strongly expressed after about week 5. That's not surprising. Those are kind of all the transcripts that are involved in being a crab. And maybe these are all the transcripts that are involved in deciding what the state of early embryogenesis is going to be. There's a lot of interesting signal transduction genes in this list. There's 137 unique annotated transcripts. That means out of that 400, we had about almost 200 that had annotations, about 50% that we know what they are. And out of those, 137 are unique, different transcripts. includes things like Hox genes that you would expect and a bunch of signal transduction genes. And they represent interesting targets for further study. Which ones of those genes are ones that are responsive to the environment? Notice these are week 1 sensitive and week 1.5 robust. And here's week 2. And in week 2, a lot of these genes continue to be expressed. So maybe like a difference in here where it's really strong in this week 1 sensitive, and here where it's really weak in this week 1.5 robust, maybe those are genes that are involved in being sensitive or not. And so we're going to do a bunch more studies to test those specific genes. And then, as I mentioned, I had a PhD student, Raphael. He was really interested in doing proteomics. So we did a bunch of proteomics on crab embryos. Never been done before, but because we have a genome sequence, we could do this. And so he discovered, comparing different lab and field samples, what the proteome is for an early embryo, a week 1 embryo, and developed this heat map to figure out, which ones were higher and lower in certain samples and which proteins are there. And he developed this network map of all the different proteins that he found to say which proteins interact with which other proteins, and there's just like tons of information. But what I wanted to know is which ones of those proteins are related to being heat resistant or heat sensitive. And that's what we see on this volcano plot right here. The y-axis tells us about how statistically different the proteins were between the 2 samples, and the x-axis tells us how different they were in terms of their expression. And usually we look for proteins that fall in these upper quadrants, either left or right. In this case, there's just 5 that are here that we would say, these yellow ones, that we would say those are statistically significant, but only barely so in this case. There really weren't strong differences. We had much stronger differences between the samples that came from the field and the samples that came from the lab than we did between the heat stressed. And actually, we did this proteomics work in a UC Davis lab, Dietmar Kultz's lab. Maybe Dietmar is on Zoom right now. And he said, I've never seen 2 samples or 2 groups of samples as similar as these 2 groups, these heat resistant and heat sensitive ones. But there are just these very few differences there. And maybe we had just a sample size that was too low. So we're going to investigate some of these proteins that were kind of marginally significant, some more, as well as this one out here that was very, very different in the heat. Actually, I don't know which side of this volcano plot is sensitive and resistant, but it was very different, but also variable enough across groups of embryos, broods of embryos, that it didn't rise to the level of statistically significant. But we're going to keep working on that. Here's the comparison of field versus lab. And so we see this is the field side over here, and that's the lab side over there. And there were a whole bunch of proteins that were expressed at a much higher level in the field than the lab, which could be due to the heterogeneity of the field compared to the lab environment. So there's some suggestion that we have changes both in the transcriptome as well as the proteome that could be additional lines of evidence for the presence of intergenerational plasticity as a mechanism for adaptation to rapid adaptation to increased heat stress. Yeah, this is just to show you the, this is 1, 2, 3, 4. I think these are some of the ones from this graph here, just to show you the types of proteins. We shouldn't really spend too much time on that. And we saw differences in a broad array of different types of molecular functions, biological processes, and cellular components. And a lot of it had to do with extracellular types of interactions, which is not surprising because there's a lot of cell-cell communication that goes on during development of the embryo. So our next steps, as I mentioned already, is to use these candidates that we've learned from our RNA-seq studies and from our proteomic studies to understand better how different these transcripts and proteins are across the different individuals within a brood, and different broods between the same parents, the same genotype, but different, a different set of embryos, and how different among embryos with broods from different parents, and how they vary in expression with development, and maybe even where these different genes and proteins are in the developing embryo. So lots of exciting things we can do on this. Okay, hopefully you're satisfied and not overwhelmed. And we'll wrap it up. So is intergenerational plasticity a thing? I think it is. I think I've shown you enough evidence to say yes, it's a thing. I hope it is. Can it be a mode of rapid adaptation to increased amounts of thermal stress? I think it can. I think it's possible that from one generation to the next, we can have changes that make the subsequent generation have a higher chance of surviving that same thermal variation. That's the Lamarckian part of this story. How does it work? We don't know yet, but now we have some tools and some clues from at a cellular level that we can investigate. Does it persist through the next generations? That is to say, does it persist through not just the embryo, but the larva and the juvenile and all the way through the adult? That's something that's completely unknown and would be really cool to investigate, although I would never try to attempt it without being at a place like Bodega Marine Lab, where you have an awesome seawater system. You don't have to worry about changing the water at a high enough frequency. Okay, I'll thank everyone who is involved in this talk and in my lab. This is a picture we took in San Francisco at a lab outing where I finally learned how to play chess from my student Joe on his last day in the lab. I should say, sorry, their last day in the lab. And also thanks to Dietmar's lab here at Davis, who helped with the proteomics work. And thank you for your attention, and I'd be happy to take your questions.
So if you have a question, we ask that you speak into the mic, which I will bring to you so that people on Zoom can hear. If you are on Zoom and have a question, I just realized about 5 minutes ago that I didn't bring my laptop, so I have no way of seeing your questions. Sorry about that, but we do appreciate you. And
Is there a way that we can stay on Zoom on this computer?
Maddie will log in to Zoom and she will monitor the chat. So if you do have a question, please type it into the chat. In the meantime, we'll start with questions in the room.
Really great. So do you think there's any epigenetic contributions to the intergenerational changes you're seeing?
Oh, can they hear me on Zoom with this one? Yeah, of course. Yeah, for sure. And that's the third possibility, is that there's changes in the epigenome that would influence which genes get turned on. And we haven't investigated that yet, but it's definitely in the plans to investigate in the future. Yeah. So we think it could be like DNA methylation, could be histone modification. Those are the two targets we'll go after first.
Thank you. Really, really interesting talk. So again, if I understood it right, you have that part where you're looking at the robust versus sensitive animals, and you really didn't see much in the proteins, right, when you compared them.
That's right.
Is that right? And so I was just curious, like, do you also have the data on like the size and the size of the yolk and stuff? Because, right, you'd shown that they were often really different.
We have the data on the size of the embryo and the size of the yolk for every embryo.
And
Yes.
so, like, were those, it just seems wild to me, like, you might have really different sizes of embryos and yolks, but maybe no significant difference in the proteins within them?
Yeah, so here we're looking for, the way that we did the proteomics was to identify the types of proteins and the relative abundance of the proteins. And the proteomics wasn't done on a per embryo analysis. It was done on a whole bunch of embryos from the brood, probably over 100, that were all homogenized together. So relating these data to an individual embryo isn't really possible, but we could relate them to the mean value of the brood from that mother. And that is a very interesting thing that we should do. Raphael, who did the proteomics work, wrote up his thesis and graduated his master's thesis, but we haven't published the data yet, but including a graph like that one would be cool to include. Thank you.
Thank you.
Great talk. I'm curious what you make of the seasonal differences in embryo size and, yolk size between spring and summer. And I'm wondering whether there's like seasonal patterns in recruitment or seasonal patterns in like population dynamics that might explain some of those differences, as well as temperature.
Yeah, probably not recruitment, but it would really be nice to know from the same genotype across seasons how different they are. So that's where we're hoping these caging experiments can work a little bit better. But the idea would be that, the idea that we have is that the environment differs across these two seasons. Temperature is different for sure, but there's probably other parameters of the environment that are also different in terms of like upwelling is really high in the spring and really low in the fall. So maybe there's different levels of calories that are being provisioned to the crabs when they're undergoing their oogenesis in the spring compared to the fall. And I don't know if we'll work on teasing all of those apart. In terms of like the population dynamics, it's not like it undergoes a wild swing from one season to the next of the reproductive adults. For sure, the recruits and the juveniles are strongly seasonal, but they're a couple years pre-reproductive. And so all of the individuals that we've studied are probably 3, 4, 5 years old. And at that point, there's not such a huge seasonal variation in their population, that demographic within the population.
So following up on Sam's question, I was wondering, so in the winter you saw that there were larger embryos that had more yolk. Is that right?
Let's just go back to be sure. This is the percent yolk and the starting size. And so for a given size of embryo, there's more yolk in the winter. But in terms of the overall sizes, maybe yes, there are some embryos that are larger in the winter, but there's also quite a good overlap.
I guess I was just wondering, in terms of the seasonal temperature variation, is there some adaptive explanation for why females might want to produce embryos with more yolk in the winter? And then I guess a follow-up question is whether, do you have information on the size of the brood, like when they're producing larger embryos, is there any classic like trade-offs between embryo size and the number of embryos in a brood?
So we have like the masses of the total mass of the brood is here. And so we also have like an average size of the embryo within the brood. So we could try to figure that out. I don't think we've done that exact analysis yet.
Questions, Jackie?
Thanks, Jonathan. I just actually have a natural history question or behavior question. Whether you notice any behavioral differences between females that are brooding and not brooding? You know, are they choosing different habitats or doing different things? Just wondering if you've seen anything like that.
Yeah, I don't have the data here to show you, but it's published, where we did like a lot of sampling to look at variation in the population demographics across their vertical distribution from the highest to the lowest that they appear in the mid-intertidal. And we definitely saw more gravid females lower on the shore than higher on the shore. And that's in a 2019 Gunderson et al. Paper called hot rocks or not so hot rocks or something like that. Yeah. But it's interesting, and if our grant to continue this work gets funded, the part of it is to have a undergraduate course associated with the research project in which they would study all sorts of interesting phenotypes on the adults that are held in the lab under these different conditions, like their respiration rates and other things, but we could do a lot of behavioral things too to see if they have different sensitivities to temperature. We've done also some like escape temperature assays and like thermal preference assays on gravid females, and there is a little bit of variation compared to non-gravid females. That's also published. I can send you those papers.
Other questions?
Another yolk-related question, more just about, so the result where there was lower yolk consumption in the heat shock broods, I was wondering, is lower yolk consumption like a signal of like worse fitness, or is it like compensating for bad conditions? Like I was like what?
I think that's it's a pretty legitimate question. Like, what do all these different phenotypic changes really mean? And we can make up some stories about it, right? Like maybe the heat shock causes a metabolic quiescence, but we're measuring their metabolic rates. We really wanted to try to characterize the whole energetics of the embryo, and we figured that they use energy that's coming from the yolk, and they spend energy on development and growth, and the rate at which that's happening should be a function of their metabolic rate or their respiration rate. And so we measured all those things, but it turns out that somehow it doesn't all add up. And it's kind of a little bit of a mystery, and not everything falls into the line that we expected it would fall, that this plus this equals that. And so there's either some problems with that idea of their overall energetics, and we're missing something there, or there's a lot of energy that's being used in a futile cycle where there's energy being used for different sorts of functions. But why, you might think if there's a heat shock that the rate of energy consumption would be higher, right? Because they have to fix the damage from the heat shock. And that is one response. But another response to heat shock is to slow down. And there's other reasons for that too, to have a quiescence that occurs. And I need to go through all of these data with a little more kind of eye towards that before we publish them all. I should say, most of what I've showed you today is not published. And this is also the first time I've ever given this talk. So never before have all these data been in one presentation at the same time before. So I really appreciate all of your ideas about thinking about how they all work and fit together.
Hey, if there are no other, go on.
Hold on, I gotta wait for the mic. Just hold on.
I'm just wondering if there is some predictive value for your results in terms of driving adaptive traits that actually would allow or enable the Petrolithes population to adapt to a warming climate.
Yeah, so I think that's like where the hatching success in the lab study was probably the one that I thought fit an adaptive story the best in terms of how the parental exposures positively influenced the offspring when the offspring also saw that same stressor as the parents did. But it fit best there, and whether it fits best also in other cases is, I think, a little more questionable. But in that case, I think our results are pretty predictive. Yeah, and it fits what you would think of from adaptive responses.
Okay, let's thank Jonathan for a really interesting seminar. And also, as promised, Jonathan, for people who are interested, will be playing the Alp horn out in the Great Hall, which has been selected for its acoustics. So give Jonathan a chance to get a drink of water and assemble his Alp horn. And then those of you who can stick around or are interested, we will meet out in the Great Hall. Thanks.